Fluorescence in Situ Hybridization, fish. Welcome to AnthroNet Academy, a study aligned with the UGC Net Anthropology syllabus. Description box. Please visit the description box for books references. Dear learners, if you find this content helpful, please consider subscribing to AnthroNet Academy. Your support really means a lot and keeps me motivated to create more valuable content for you. Big surprise at 500 subs. Once AnthroNet Academy hits 500 subscribers, we're dropping something truly special just for you. A full video revealing 100 powerful, underrated, and must-read books listed in Anthropology, complete with author names and insights important for UGC NET exam. Introduction. The classical cytogenetics used trypsin gamma, or fluorescent banding pattern, for identification and characterization of different chromosomal abnormalities, such as polycentric chromosomes, ring chromosomes, or chromatid interchanges. Fluorescence in situ hybridization, FISH, is the most convincing technique for locating the specific DNA sequences, diagnosis of genetic diseases, gene mapping, and identification of novel oncogenes, or genetic aberrations contributing to various types of cancers. Advancement in FISH techniques. Fluorescence in situ hybridization, FISH, can detect specific sites of specific DNA sequences in metaphase or interphase cells. This technique, initially developed for mammalian chromosome, was first applied to plant chromosomes by Schwarzacher et al. 1989, and Yamamoto and Mukai, 1989. FISH has been used to detect 18S26S rRNA and repeated DNA sequences in plant chromosomes such as Aegilops, Hordeum, Oryza, Arabidopsis, Brassica, soybean, and barley chromosome. Core principle. Nucleic acid hybridization. A fluorescently labeled DNA or RNA probe binds to its complementary sequence in the sample. Detection is done by fluorescence microscopy to visualize location, number, or arrangement of target sequences. Step-by-step procedure. Sample preparation. Tissues, cells, or chromosomes are fixed on slides. Denaturation, with heat or chemicals, is used to separate DNA strands. Probe preparation. DNA or RNA sequences labeled with fluorochromes, e.g. FITC Cy3 Texas Red. Probes are designed to complement target sequences, e.g. gene chromosome region. Hybridization. Probes are applied to the denatured sample. Under hybridization conditions, typically overnight, at 37 degrees C, the probe binds to the complementary DNA. Post-hybridization washes removes unbound or non-specifically bound probes. Ensures specificity. Detection and imaging. A fluorescence microscope is used to visualize. Multiple probes can be used simultaneously, with different fluorochromes, multicolor FISH. Diversification of FISH techniques. Widespread recognition of FISH as a physical mapping technique to support massive nucleotide sequencing is involved in the Human Genome Project. It has become a more convenient and popular technique in other areas of biological and medical research, including clinical genetics, neuroscience, reproductive medicine, cellular genomics, and chromosome biology. FISH technique. Some of the techniques listed below, which have been inspired by the glossary of Volpe and Bridger. ARM FISH. ARM FISH is a 42-color M-FISH variant that allows the detection of chromosomal abnormalities in the P and Q arms of all 24 human chromosomes, except the P arm of the Y and acrocentric chromosomes. ACM FISH. ACM FISH is a multicolor FISH assay for detection of chromosomal abnormalities in sperm cells. The abbreviation ACM refers to the simultaneous hybridization of DNA probes for the alpha centromere, classical 1q12 satellite, and mighty 1p36.3 satellite of chromosome 1 for the specific detection of duplications and deletions of 1p teacher and 1e cin, and for the identification of chromosomal breaks within the 1cin1q12 region in human sperm. The discovery of chromosomal break or damage in the human sperm provided explanation for infertility in oligozoospermic men. Catalyzed reporter deposition fish, cardfish. Cardfish refers to the fluorescein tyramine signal amplification, mediated by horseradish peroxidase, HRP, labeled oligonucleotide probe. This technique is very useful for detection, identification, and quantification of microorganisms involved in bioleaching processes. Cellular compartment analysis of temporal, CAT, activity by fish, catfish. Catfish uses fish to immediate early RNA genes and confocal microscopy to identify neuronal population activated at two distinct times. This technique is used to determine the interactions of neuronal populations associated with different behaviors. Cytochalasin B, CB fish. CB fish involves hybridization on binucleated cells in which cytokinesis has been blocked by treatment with cytochalasin B, CB. Shows increased ratio of mosaic diploid cells in vivo in trisomy 21 cases. Analysis of the chromosomal content of micronuclei can be facilitated by combining the standard CB fish protocol with the 24-color sky technology. Chromosome orientation, CO, fish. Co-fish uses single-stranded DNA probes labeled with 5-bromodeoxyuridine during S phase to produce strand-specific hybridization. It allows to determine the relative orientation of two or more DNA sequences along a chromosome. Initially, this technique was designed to determine the orientation of tandem repeats within centromeric regions of chromosomes. This technique has also been useful in assessing chromosomal translocations and inversions. Combined binary ratio, cobra fish. Cobra fish enables recognition of all human chromosome arms on the basis of color and mapping of gene and viral integration site in the context of chromosome arm painting. Cobra fish protocol brings together combinatorial labeling, which allows different ratios of label to distinguish between probes. This permits the use of fewer fluorochromes to produce up to 48 color combinations for differential painting of human chromosome arms within a specimen. Chromosome orientation and direction, COD, fish. This protocol is similar to co-fish, except for the information about the directional organization of telomeric sequences. It can also stand for concomitant oncoprotein detection fish, which allows visualization of loci signals for a particular oncogene and also the protein product derived from this gene. Another technique that has been termed codfish is the combined KKO3 optical detection fish, in which fish is used to detect calcifying microorganisms in open ocean. Combinatorial oligonucleotide, combo, fish. Combo fish is used for specific labeling of genomic sites. It takes advantage of homopurin/homopyrimidine oligonucleotides that form triple helices with intact duplex genomic DNA. This will not require prior denaturation of the target sequence, which is usually a prerequisite for probe binding in the standard fish protocols. Homopurin or homopyrimidine regions of DNA are usually longer than 14 BP, representing 1 to 2% of the human genome, with an average of 200 of such stretches in a 250 kilo segment of the genome. Accordingly, specific probe sets can be constructed to target genomic regions of interest in that size range. Applications of fish. Area. Application. Cytogenetics. Detect chromosomal abnormalities, e.g., deletions, duplications, translocations. Oncology. Identify gene amplifications, e.g., HER2 in breast cancer, translocations, e.g., BCR-ABL and CML. Prenatal diagnosis. Detect aneuploidies, e.g., trisomy 21. Microbiology. Identify and localize bacteria or viruses in clinical samples. Comparative genomics. Map genes across species. Advantages versus limitations. Advantages. High specificity and sensitivity. Spatial resolution of nucleic acids within cells. Can detect submicroscopic changes. Useful in interphase nuclei. No need for metaphase. Limitations. Requires prior sequence knowledge for probe design. Can be time-consuming. Expensive compared to PCR or qPCR. Lower throughput compared to NGS. Thank you for watching. Please like, share, and subscribe to my YouTube channel, AnthroNet Academy, for more insightful content on anthropology. Stay safe and connected.